we can talk about the human genome. And so that's all the DNA that is in your cell, and most of it is located in the nucleus. So this is all the DNA in a cell, most located in the nucleus. One place where it's not located, or where it is also located, is in your mitochondria, but most of it's located in the nucleus. So this human genome is 3 billion base pairs. So what does that mean? That means that our DNA is composed of nucleotides, and I'm sure you have heard of the letters. So this would be like A, G, T, C. Those are abbreviations for adenine, guanine, thymine, and cytosine. You do not need to know those yet. We're going to talk about those in more detail when we get to the part on the DNA molecule. But these nucleotides, our DNA is double-stranded, so it forms base pairs. So the A, G, T, and C are actually the bases. So when we look at a sequence, we could write it like this. This is one strand. The other strand, the A's always pair up with T. The T's pair up with A. This is called Chargaff's rule, which we'll talk about in more detail. But I just wanted to show you what a base pair is. So this would be one base pair. So this is one base pair. So this is a base. Ooh, that's a bad color. Let's see. Better color. This is a base pair. Okay. So we have 3 billion of those. So that means we have 6 billion nucleotides in our genome. That is a lot of DNA. The crazy thing about our genetic material is that only about 1% of the genome actually codes for protein. So about 1% of genome codes for protein. And so when we look at what that means, when we talk about genes, genes are pieces of DNA that code for protein. And you might have heard of the Human Genome Project, which took place in the 90s and the 2000s, where they were trying to sequence the genome. So sequencing would be the sequence of A's, T's, C's, and G's in your genome, but they were also looking for where we might have genes in our genome. And what they had discovered is that there was actually not that many genes. So there's only about this many genes in our genome. So the question is, what is all that extra DNA doing, right? What is 99% of the DNA used for? And we will talk more about that when we get to molecular genetics.